So now let's move towards the degradation of pyrimidines. The degradation of pyrimidines is not as complex a process as was the degradation of purines, because in purines you have to deal with double ring compounds, while pyrimidines are consisted of single base rings. And also one other thing you have to keep in mind, that it doesn't have to be excreted in the form of the degradation products of these compounds doesn't have to be excreted in the form of uric acid. These are highly soluble compounds, their degradation products, or even the pyrimidines themselves are highly soluble, and they can easily be then dealt with through the degradation process. Degradation of CMP, that is cytosine monophosphate, and UMP, uridine monophosphate, they produces simple amino acid like beta aniline, which is again highly soluble product, which makes it less available for the salvage process and more easily to get rid with, because their degradation is much more easier. Then the degradation of the third base, that is our third type of nucleoside, pyrimidine based nucleoside, the TMP, the thymidine monophosphate, it produces beta amino isobutyrate. Beta amino isobutyrate, beta alanine, they are very highly soluble, and hence to get rid of them or to deal with their degradation is quite different as compared to dealing with the purine degradation. The byproducts of these degradations are ammonia and carbon dioxide. Now, the ammonia here, as in the case of purines, you saw that the byproduct of the degradation, one of the final product which has to be excreted through the kidneys, was the uric acid. Here it is ammonia, and carbon dioxide, of course, you know the root of the carbon dioxide, and you have already studied how ammonia is excreted from the body. Ammonia, keep in mind, is highly toxic, but thankfully, we have a very, very well-defined way of getting rid of ammonia in the form of urea cycle. This ammonia becomes part of the urea cycle, and then it is excreted in the form of uric acid. So, keeping the things simple, I have already told you that keeping the things simple, we can somehow salvage or use this pyrimidine metabolism for the salvage process, in which you make more pyrimidine molecules from the already present pyrimidine bases. But because of their high solubility, they are not that much available for the process of salvage process. This was not the case in the purine. So, and also you can think of that in the case of purines, you had to make double ring compounds, which are more difficult to make as compared to very simple, relatively simple pyrimidine compounds. So you don't have to depend upon salvage process for the formation of pyrimidine based bases or nucleosides. So that's one of the things you have to keep in mind. Thank you.